Okay. Well, thank you. This is work with Edine and other folks here in the group, BJ and Shweta, to talk about our AIM-5. The details were to implement uh, an API for annotating and filtering samples in single-cell experiments, and also to deal with minting and defining new ontology components to define new classes of cells. I think it's worth reflecting on the idea that the cell ontology exists and gives mechanisms for naming and tagging cell types. But these tags are infrequently used. Certainly in the tutorials that we have, it's very seldom the case that you get an exact reference to a named cell type. And we think that we should do a better job here because we want to implement the FAIR principles to make things findable and interoperable. And the problem is that, especially when you're at the boundaries of knowledge, people doing new types of identification, it's not always clear what you want to say in terms of an ontology. So here's a case of the mature eosinophil. It's important for us in lung disease research. And you can see that the definition here involves a great many potential features. And we want to be able to systematically handle this type of identification. On the other hand, the question is, how are you going to refer to these features here? Are these parts of the cell? These are phenomena? You take something like has plasma membrane part or lacks plasma membrane part, an important part of cell identification. Should it be a subproperty of lacks part? And there's actually a dispute about this that ranges over multiple years. And you can see that this was assigned to a group called Gene Ontology Cell XP, but that project went stale a couple of years ago. So what we've tried to do is produce tools that help people work with what exists in cell ontology. And I'll show you an app called CT Marks that helps work through multiple ontologies to work with existing definitions of cell types. And then we come up with a protocol for how we would put new information in and bind it to ontology terms and allow people to work with these as they try to annotate their data. So here's a nice use case, kind of an unusual one, I think. We have a set of genes here, and it turns out, this is work done by Richard Scheuermann's group, and he is one of the ontology leaders in the HCA. For this gene set, there's a certain collection of cell types, they believe, where exactly one of each of these genes is expressed by each of these cell types, and all the other genes are not expressed. So I call that a diagonal pattern or diagonal signature type. And what we want to do is incorporate this type of information. It's not available in the cell ontology. And what we want to have is an app that will enable people to look through different cell types. In this case, there's 400 or so cell types that you can look at through the app to find out what cell ontology says about them. And if they aren't already in there, you can create some new cell ontology classes and have conditions such as has expression of GRIC3 or lacks expression of the other genes in this diagonal pattern. So that's been done in the CT Marks app. And this just goes into how you would set up the data for import. Let's say you just make a little table. You have the genes, you have the type you're trying to annotate, and whether or not the gene is expressed. Now, we may want to have other conditions identified here, but we're just working with these two simple ones to begin with. And these terms, has expression of and lacks expression of, these are proposed in Scheuermann's papers, but they have not been adopted yet in the cell ontology as far as I can tell. And the tools for getting together so that we can get into this app, which I think I'll just show you folks now. That's here. It's in shinyapps.io. We can reload, and we'll just get a feel for what is in there. So you can pick the cell type that you're interested in. let's say I was interested in that GRIC3 expressing interneuron, then you click on that, you get a sense for how it is derived through the hierarchy, the genes that are involved and the expression and so forth. And this other thing tells you about the actual ontology data that underlies this app. That's actually pretty substantial. Cell ontology makes a lot of use of protein ontology in order to annotate the features, and that itself is heavily curated out of many other ontologies. So, to conclude, what we want to do is support the FAIR principles for de novo cell type definition and allow people to experiment with formal annotation before, let's say, it gets adopted by the EBI as part of cell ontology. Ontology development is very regimented. As I showed you, there are disagreements, and you need to really think about standards compliance. There's something called Webulus at EBI that will help generate OWL-compliant ontology content, but it doesn't cover the types of problems that we're dealing with here. And we'll keep working on this to try to curate feature sets and so forth. And we need to think, I think, about a signature server so that people who are looking for signatures to try to interpret their new data can get them easily, and then also push information that they find back into the system so that we're all collaborating as efficiently as possible. So that's the end of my presentation, and I'll stop sharing.
Thanks very much, Vince, and that's the end of the presentation part. Thanks very much to everyone for the tremendous amount of work that's been involved in the developments that have been summarized, and also for participating in the virtual symposium today. It's totally great and really informative to see all of the work that's going on.